Okay, lecture 11, which I will do live, but this is for anyone who sort of misses the live version of this. So we're going to look at what causes food spoilage, what it is, source of it, the very important issue of biofilms, the tragic news about the best website ever, and some new technology approaches. So what is food spoilage? Always good to start with a definition. And here's the definition. You can read it yourself. Food changes naturally with time unless you deep freeze it. In some cases, such as with this blue cheese, this adds flavour and is desirable. Often though, the changes make the food uneatable and possibly dangerous to eat. This is referred to as food spoilage. We'll note that blue cheese contains colonies of blue-green mold penicillium, which produces distinct taste and smell which many people like and many people don't like. Okay, so signs of food spoilage. Okay, bring that up. I'm quite sure why that wasn't bringing up. These obviously some bread which is having some problems. Signs of food spoilage include changes in colour, texture and odour and taste. And mould is often clearly visible on the surface of the food item. And the point about that is, I guess, it's not just bacteria, it's often fungi as well, microfungi and moulds. So, putrefaction. Bacterial putrefaction produces unpleasant smells and tastes in food. Often happens under anaerobic conditions. There's just some definitions of what the words mean. The process involves the breaking of peptide bonds in proteins to produce amino acids. Further decomposition leads to production of ammonia and hydrogen sulfide, all of which gives the unpleasant smell, which we perceive as putrefaction. So fermentation. A wide variety of microbes produce enzymes that can produce fermentation. Fermentation, of course, is the growth of microorganisms and foods, of which foods present a good environment for. Water activity issues are relevant here, of course. One example of this is the fermentation of milk, which converts lactase to lactic acid, such as in yogurts. And here's an example of the fermentation process. Here's a summary of some of the potential negative effects of food spoilage on food quality. You can pause the video and have a read of this yourself. This, of course, is covered in more detail in the food safety module. Just for a definition there, biogenic amines are organic bases of a low molecular weight and are synthesized by microbial, vegetable or animal metabolisms. In food and beverages, they are often formed by the enzymes of raw materials or are generated by microbial decarboxylation of amino acids. So again, another decomposition process. So they're the microbial causes, but there are sort of reasons why it happens, which is what we mean by this. Physical, biological and chemical. We can classify them into these three classes. You should think about them. It says they're in groups, but you can do this on your own. You can get together in groups and talk about this. You can think about the causes of each classes of food spoilage before advancing the presentation. Pause the video if we're watching the video. Okay, so here's a mind map sort of summarising food poisonings food spoilage causes, I beg your pardon. Contamination can be caused by lack of control of the physical environment of food, both in storage and transport. For example, the cold chain may have been compromised. Microbial contamination may be caused by microbes that exist in the general environment or from various types of contamination. These two issues are discussed here, but again will be reviewed in more detail in the food safety module. The chemical causes include enzymic ones, which would include the action of polyphenol oxidase, and oxidative damage to lipids, for example, which we will look at later. Okay, so sources of microbial. I've got this in twice, I haven't. Yeah, factors. There should be factors affecting microbial spoilage. Ignore the previous slide. Again, some definitions here. Extrinsic means factors outside the food itself. For example, the temperature at which a food is stored is an extrinsic factor. Intrinsic factors are those associated with the food itself. So, for example, its water activity or its pH. Process activity issues are outside the scope of this module and will be discussed in other modules. In this case, the word dynamics refers to issues such as interactions between microbes, especially in surfaces, and more on this in a moment, and the importance of understanding microbial growth rates. Again, you will cover this in other modules. Now, the point about this is that food poisoning organisms are all over the place. They are said to be ubiquitous. Those of you who haven't come across the word ubiquitous means something that can be found almost everywhere. For example, people are ubiquitous on the planet Earth. Natural habitats of microbes, for example, is the soil, and is said to be ubiquitous there. Apart from pathogenic bacteria, which the natural environment, of course, is living organisms. Food can be contaminated directly by contamination with soil or indirectly by water and air. Also be present on skin, both human and animal, plants, insects and surfaces in general. So there's lots of sources of potential contamination for food in the environment is the point of this. If food is damaged in some way, this allows microbes to access the deep tissue, increasing the amount of contamination. If food waste is not segregated from ingredients in use, this again can often lead to contamination. Okay, so biofilms. How are we doing for time? Yeah, okay, so we'll continue this one. Read this article for more information. It is linked through, but you'll have to log in via the university account. It's in the background. It's a sort of background reading for the module, so you should have a read of it. It's published in 2013, so you might want to look for a more recent article, of which there are many. The biofilms are a really important area, and there's an interesting picture there from a scanning electron microscope of E. coli on a lettuce leaf. Point about this being there is issues with food that isn't cooked, which salads are obviously one of the classic examples. Okay, so again some definitions. A biofilm is a group of microbes going together on a surface. They often exist in the slimy matrix called the extracellular polymeric substance, EPS. Technically, not everything that feels slimy is a biofilm, but many are. Biofilms come from on living and non-living surfaces, so equipment, benches, door handles, etc. may be contaminated in this way. The fact that the microbes are living together often makes them resistant to antimicrobial agents, and they may also promote the corrosion of equipment. Later on we'll look at a couple of new methods for dealing with this issue. There's a good definition there from our IU pack on what a biofilm is. Here's just an example. Cyanorhosium milioti is a gram-negative bacterium which fixes atmospheric nitrogen. It forms a symbiotic relationship with various legumes. No need about that level of details. I just include this as an example EPS. We'll notice it's a polysaccharide. Fungi as well as bacteria can also produce biofilms. Candida albicans is an opportunist pathogenic use. It's a common member of the human gut flora. It is also found in food. Again, a scanning electron microscope image of a C. albicans biofilm. Okay, so how biofilms form. There's an innocent surface there, and cells land on it and adhere by weak and reversible van der Waals forces. Cells start to produce EPS. This adheres the molecules more firmly to the surface. As the film matures, colonies start to form. In stage 4, the biofilm becomes a 3D structure with channels joining clusters of cells. It becomes fairly obvious that it's there at this stage. In stage 5, the biofilm disperses to form new cells, disperses cells that can form new biofilms. Okay, so back to exopolysaccharides, slime. These are the polysaccharides that can be produced and exuded by bacteria and fungi outside themselves in the environment. These EPS are very diverse in composition. They often contain high molecular weight sugar polysaccharides, some of which have industrial uses. They have many uses in food. Now we saw on the previous slide a mature biofilm. In mature biofilms, cell division is uncommon, with cells tending to use energy to produce extra polysaccharides, which the cells themselves use as nutrients. Here's an example of an industrial important one. Dextrin EPS is produced by several bacterial species, and dental plaque is often also quite high in this substance. It's produced industrially by lactic acid bacteria. Dextrin is one of the first microbial polysaccharides to be commercialized and to be approved for use in food. And here's some examples. Food applications include bakery products, confectionery, ice cream, reduced fat cheeses, frozen and dry foods. EPS, synthesized by lactic acid bacteria, play a major role in the manufacture of fermented dairy products such as yogurt, drinking yogurt, cheese, fermented cheese, milk-based desserts. And some of the examples here, if you read this paper, there's it's a good review of applications, and it's quite an old paper. Again, you can probably find something newer than this. We mentioned Nordic ropy milk, sometimes called viili. This arises from low temperature fermentation, not low temperature fermentation, it says in the notes, and produces a stiff, viscous product. It's characterized by ropy consistency caused by slime, that is EPS, excreted into the milk during the exponential growth phase of the microflora. And again, there's a link there which we'll talk about more about these types of products if you're interested. And there's a picture of a Nordic chap, hopefully making ropy milk. Gone but not forgetting. The Nordic Food Lab was the best thing in the lab, covering all manner of strange things eaten by our Scandinavian friends, including tree bark, flour, multiple stages of rancid butter, squeaky cheese, and lots of others. It's now been subsumed into a University of Copenhagen archive, which isn't as well presented, I don't think. You can download the whole archive from this link here. It's a 487-page PDF, which I will muse over when I have time. It's nice, but it's not the same, which leaves the previous world's best, the best thing in the web, the World Carrot Museum, having retained its previously adopted crown. Some good stuff on the University of Copenhagen website, it has to be said. Okay, so new approaches to slime. The persistence of foodborne pathogens on food contact surfaces and food processing lines is a matter of concern for public health organisations and food industries. Every year, 600 million people are afflicted with a foodborne illness, resulting in 420,000 deaths according to WHO data. So cleaning obviously remains very important, but a range of new approaches are coming into use to control bacterial growth leading into slime formation, which, as we mentioned, can be a bit resistant to current techniques. We'll look at just two new approaches here. Search the literature for more if you're interested. Yes, 2021, so a brand new paper just been published. Flavozyme is a food-grade peptidase enzyme from Aspergillus oryzae, also known as koji mold, a filamentous fungus used in Japan to saccharify, that is, add sugars to rice, to reduce them to sugars, rice, sweet potato and barley in the making of alcoholic beverages such as sake. It also mentions UHMWPE, that stands for ultra high molecular weight polyethylene. So the experiments on this were done on UHMWPE and rubber surfaces. DNA hydrolase, which is a peptidase which hydrolyzes DNA, is often used, whereas the peptidase used here hydrolyzes proteins. And the paper reported a significant reduction in young, 24-hour-old to mature, 72-hour-old biofilms on both surfaces after treatments with Flavozyme. And the overall reduction potential of the Flavozyme was a bit higher than that of the often used DNase enzyme. So an interesting approach there. And again, there's the paper there if you want to have a read of it. Something that's becoming useful is use of cold plasma. And this is a specific application talking about structures that are produced by 3D printing. Cold plasma is partially ionized gas composing of ions, electrons, ultraviolet photons, and reactive neural compounds such as radicals, excited and ground state molecules. Cold here refers to temperatures of about 450 degrees C compared to 1,000 degrees centigrade of normal plasma. And the paper here considered the role of cold plasma inducing the growth of bacterial films on 3D printed food handling implements designed for people with mobility issues, or food handling issues. The 3D printing process tends to promote bacterial adhesion and biofilm formation. Here the process involved polymerization of plastic coating with cold plasma. And again, you can read the paper if you're interested. Paper's over there. The paper reported that acrylic acid plasma coatings suitable for decreasing the amount of biofilm generated by different bacteria over 3D printed structures, as is shown here. So this is obviously another one of those examples which looks at food technology but links through to areas such as dietetics. So, yep. Okay, and you can also see this article. That's what it looks like, the purple blowtorch that blasts the blues, in this case the blues being food poisoning microorganisms. Okay, so there we are. That's all for this. Thanks for listening.